Hi, my name is Takeshi Shimi. I'm a cell biologist studying at the World Research Hub Initiative and Institute of Innovative Research, Tokyo Institute of Technology. Let me introduce my research project. DNA is a material that encodes genetic information, including the codes for proteins. In eukaryotic cells, DNA is folded into a high-order structure and enclosed with the nuclear envelope as stored in a place called as the nucleus. In the mammalian cell nuclei, the nuclear lamina lines the nuclear envelope to maintain the nuclear structure, while the nuclear pore complex is a tunnel penetrating the nuclear envelope for macromolecular traffic between the nucleus and the cytoplasm. Immediately after the messenger RNAs are transcribed from the DNA inside the nucleus, they are transported to the cytoplasm through the nuclear pore complexes for translation of proteins. These discoveries have led to the idea that the lamina and the nuclear pore complex have distinct structures not only to regulate the transcription, but also to control DNA replication and repair. The functional aspect of the lamina and the nuclear pore complex are further supported by the observation that the temporal changes in the structure and the distribution of the lamina and the nuclear pore complex are in fact among the most significant hallmarks of differentiation, aging, and cancer. However, the molecular mechanisms have not been fully understood yet. Our long-term research goal is to have a better understanding of the structure and function of the lamina and nuclear pore complexes. The major structural components of the lamina are type 5 intermediate filament proteins, nuclear lamins. Lamins are subdivided into the A and B type lamins. Over 500 mutations in laminae genes cause a spectrum of human genetic disorders, collectively termed as laminopathies. Symptoms caused by a large number of lamin A genes are mostly commonly associated with the dilated cardiomyopathy and the skeletal muscle dystrophy. Our previous studies revealed that lamin molecules actually assembled into a filament with a diameter of about 3.5 nanometer, and that these filaments non-randomly lie on the top of each other to form meshwork structures with the thickness of about 13.5 nanometer. On the other hand, the nuclear pore complex is known to be a supramolecular structure that has an overall cylindrical shape with a diameter of 110 nanometer and contains more than 30 proteins known as nucleoproteins. We recently found that lamin filaments and the nuclear pore complexes were in close proximity to each other and acted together to maintain their structure and distribution. Our current research focus is to study nuclear functions for maintaining the structure and distributions of the lamina and nuclear pore complexes under mechanical stress. We hope that our future findings will shed light on the physiological property of and the pathological changes in the laminopathies. Last but not the least, it is my honor to be a member of the World Research Hub Initiative and the Institute of Innovative Research, Tokyo Institute of Technology. This provides us great opportunities for working and discussing with the outstanding scientists and wonderful colleagues from all over the world. We have on-campus access to open research facilities for life science and technology, equipped with a variety of state-of-the-art instruments according to our specific needs of the experiments. Especially, I can't emphasize enough the significance of the cutting-edge technologies available in our institute. For example, obtaining imaging probes for microscopy, including FibroM and MintBody, from the laboratory of Dr. Hiroshi Kimura is extremely beneficial for conducting my own research. Thank you so much for your attention.